Hi everyone, welcome to the Biotech session. I'm the moderator, Linya Wang. I'm currently working in Twist Bioscience, which is a synthetic biology company. We're utilizing different libraries for antibody discovery. So before we start this session, we wanted to know how many of you are working in biotech. Please raise your hand. Okay, thank you. So now our speakers have a brief idea. So before we start to ask questions, I'll let the speakers to introduce themselves first. So let's start from Leon.
Hi, everyone. My name is Leon Huang. Before I introduce myself, I would like to thank for Leah actually invite me to this forum and also the committee of the summit. Give me this opportunity to come here. I live in San Diego, actually. I flew here and then had the opportunity to share my experience. So I'm currently a director of immuno-oncology discovery at Pfizer. And I'm leading a group of 18 scientists, including 9 PhD, responsible to develop novel immuno-oncology therapeutics, starting from scratch, from target discovery, all the way to modality generation, identification screening, and then IND filing and first in human clinical trials. So my group pretty much covered the early discovery all the way to phase 1 trials. And that's one of my current responsibility and job function at Pfizer. I have another responsibility and job, which is associate with Northwestern University. Currently, I'm the adjunct associate professor at Northwestern University, where I collaborate with the faculties in the pharmacology development and help and then mentor their immuno-oncology research, including manuscript preparation and also RO1 grant submission. So, I kind of had both flavor of industry and also academia discovery research currently ongoing simultaneously. And I found that is a very fulfilling experience I currently have, and I wish I can provide more insight and experience with everyone. That's my brief introduction. Thank you.
Thank you. And I-Chueh?
Hi, thank you for the chance for me to participate in this symposium. So, I'm I-Chueh Huang. So I'm currently working for Johnson Johnson in the gene therapy department and a principal scientist. So currently I'm focusing on develop different kind of new platform for gene delivery. And also one important part for my work is to find the immune mitigation strategy. So for gene therapy, we don't want immune response, totally different from oncology. So we need to consider from the opposite side, yeah. So before joining J&J, I did my medical degree in Taiwan, National Taiwan University, and I also practiced there for 3 years in internal medicine. And then I did my PhD and the postdoc in Harvard University, and so my focus was virology. So I stayed in academia for about 8 years. So, and then I jumped to the industry. So I think it's highly possible because of my medical background. So I really want to do something more relevant to patients. That's why I joined the industry.
And Andrew?
Yeah, thank you for inviting me here. It's an honor. So my background is actually pretty broad. My undergrad major was in physics. So people who study physics, they usually don't like biology. I didn't like biology. But in my third year of college, a professor introduced me to use physics to study protein structure. And then you can use physics to study protein-protein interaction. This is not simulation. You can actually use microscope to look at protein and protein interaction, the dynamic. And I thought that's really cool. You can apply physics to study biology and to study disease. So I decided to pursue my biophysics PhD. So I went to Michigan, University of Michigan for biophysics, where I did a lot of things. I was building microscopes, so I need to know some mechanical engineering, so I need to mix a machine so that I can put some sample onto the machine. I need to write software to control the device. I need to write software to analyze a lot of images I took. And then I had to learn how to handle mouse, because I was doing some Alzheimer's disease study, and we need to culture some neuronal cells. So I did a little bit of everything. After that, I went to Genentech for my postdoc. The initial goal was to become like the expert in imaging, no matter it's MRI, PET, or basic microscopy. But long story short, I discovered my true interest, which is protein engineering. I didn't know what was protein engineering at the time, but I fall in love with it. And also the combination of that with machine learning, especially natural language processing, or people call it nowadays language model. So that two combination make me fall in love with this field. And long story short, 2019, I think my idea is pretty mature. And it's not just an algorithm, it's a process of generating data, power the language model, learn, predict, and then validate, and then generate another set of the data. That feedback loop is really powerful. And I think if I stay at big company, this technology cannot really benefit other companies. So I quit my job to form this company. But I didn't have any experience building companies, so I instead joined a few startups. So I joined a Series A company that was doing protein engineering. And then I joined a Series B company that was doing cell-free DNA diagnostic, and then a Series C company that was doing CRISPR, gene editing. And then this year, I think my company is pretty mature, and the market is also very mature. So I decided to focus on my own company full-time now. Thank you.
Thank you for the background introduction. So first, I would like to start with personal journey part. So for Leon, I would like to know that after your graduation, you mostly joined a big pharma. Can you talk to us about like resources, why you prefer big pharma? How about the collaboration, the eyesight? Is that the part why you always choose big pharma?
Yeah, so I need to go back to my graduate school study first. And I think that intrigued me to pursue my career in industry, particularly big pharma later on. So when I was a grad student at UCLA with immunology major, I happened to have the opportunity to invent immunotherapeutics called cytokine antibody fusion, and then licensed to a small company for clinical development. And then, I witnessed the whole process from the start of the idea, inventions, and then licensed to the companies and pushed to the phase 1 trial. The struggles, the excitement, everything in the whole journey. And then, although the drug pushed to the phase 1, but however, eventually the company cannot continue the drug development beyond phase 1 because they lack of funding, because they are a small company, they don't have sufficient funding. And then at that time, the cytokine antibody fusion is a very novel idea. At least there's no FDA-approved cytokine antibody fusion yet for immunotherapeutics. So they have struggled to secure enough resource to push the drug forward. So I realized the challenge working in a small company, because that could be the challenge I was facing if I chose a small company. So after my postdoc training, I pretty much determined I want to go to industry, because with my experience in drug development and immuno-oncology. And then, I applied multiple positions across small company and mid-sized company to large-sized company. And then, fortunately, I got interview offer for all the companies. And then, even though, I'm pretty sure that I don't want to go to small, mid-sized company, I still go for interview for the purpose to understand what they are doing, and also understand their culture, and also understand the way, the difference between big pharma and small company to execute, to initiate the program, how they move the drug program forward. So from there, pretty much see that actually in the big pharma, you can see the whole portfolios, different diversity of the drug development program that impact the different aspects of unmet medical need in patients. And small company, most of the time, they can only focus on 1 or 2 areas, or one or two targets at most. So, I feel like for my career development, if I put myself into big pharma environments, I'm able to learn more, actually see the whole picture. That's definitely more beneficial to put into the more narrow scope environments that my experience will be limited in that kind of setting. So that's why I pretty much determined to stay in big pharma. And then, as you work in big pharma, you get more visibility, more experience, there's more opportunity, including internally, externally coming up. And then, that's how you grow your career. And then, I've been taking that advantage and identify even more better opportunity in other pharma. So, my career started at Amgen as a senior scientist. And then later on, I have the opportunity to move to AbbVie as a group leader to lead a group of multiple PhD. And now I grow into a director role position at Pfizer, leading the much bigger group, and also supervise multiple drug development programs, facing from very early stage all the way to phase 1. So I feel this journey is very fulfilling, and also I get a lot of experience, which I don't think I can get similar experience if I start with in a small company.
I see. And then for I-Chueh, you have a very interesting resume. You start with MD by training, and then you go to USC, where I know you as a professor, and now you join industry. So can you share with us how you make this decision and compare different experience?
That's a good question. So what there was in National Taiwan University Hospital, so there was a SARS outbreak. Yeah, it's probably the main cause for me to decide to do a PhD because I have the curiosity about mechanism, about the mechanism. So, and I applied the school and fortunately get into Harvard, and also a turning point because when I grew up, so I need to listen to my father. So for whatever reason, I did the interview for my father. And then I have a chance to get out to do what I really want to do. So that's why I changed to the biology field and start to do research. And in my research career, the good thing is you can do a lot of things you are interested. There is no limitation. And but the thing is, if you run your own lab, the resource is quite limited, and then you pretty much need to do everything by your own. And then even you can find some collaborators, so they do have different priority. So sometimes you feel a little bit exhausted in the academic field, and it's a highly intensive working environment, I would say. And when you stay in academic for a long time, you do very detailed molecular pathway or very specific mechanistic studies. So sometimes you just feel you lost the whole forest. You don't know the potential clinical application of your research. So that's the difficult part for me. That's why I think probably working in the industry can give you a better opportunity to redo something more relevant to patients. It's why I decided to change my career and luckily get into J&J. So, and in the industry, you do have a lot of resources. Yeah, and you don't need to do everything by your own. So you can easily find expertise, different department, especially for big pharma. Yeah, so it's the thing. I think I did the right choice. So yeah, I'm happy for working for my current position.
Thank you. And for Andrew, actually, you already answered why you start your own company. So my question is for you is that why is it AI? And why do you focus on protein? What application are you working on?
Right. Maybe I'll start with why I decided to focus on this. As I mentioned, my training was in physics, optics, and microscopy. So my first postdoc project, also my last project, is to look at T cell, be able to visualize T cell in human body, take a bunch of images, and then predict the T cell concentration around the tumor. And that allows us to estimate how well genetics immunotherapy works. And at the time, I think Sonny also mentioned, we have a lot of antibody that can bind to T cell. But the problem for us is the antibody is very good for making drugs because they circulate in the body for a really long time. But as an imaging purposes, we don't want that long circulating time to interfere the signal. So what we want is a protein that can quickly bind to the T cell. We take a bunch of images and then fall off and cleared by the body. So, and we don't have any protein that can do that. That's why I got into protein engineering to try to search for that protein. And I didn't know what protein engineering mean at the time. And you can think of like software engineering. Software is that you are writing computer code to ask the computer to do something. Protein engineering is, if you don't, can I ask how many people know what protein is? Or you don't, if you don't, okay, good. I didn't know. I mean, I thought protein is like chicken or egg, you have protein. But it turns out, so protein is a sequence of amino acids. It's like a human language without white spaces, so just characters. And each character have 20-ish different amino acids, A, B, C, D, E, F, G. So what you wanted to do in protein engineering is that you are manipulating those amino acids. You hope that this protein will do something for you. So that's protein engineering. And turns out there's a really cool technology that you can generate 10 to the 11th different proteins. So that's pretty big number, and that's a big data set for doing data analysis. And this is not simulation. This is actually in the lab. You can ask a virus to produce a lot of protein, different protein you want, and then you do measurement. Now, the problem there is at the time, 2014, when I joined the company, you can generate 10 to the 11th, but you can only look at few hundred and then pick from few hundred which one can bind to the target. So that's you have this many data as input, but you're only looking at this. And I thought this must be a better way that can look at much bigger data. And at the time, NGS, next-gen sequencing, is another very cool invention in the industry, and I love it. That technology is being used to study disease, study how gene mutation affect the disease, but not a lot of people use that in protein engineering. So I kind of introduced that technology into the company, and I wrote a lot of software to analyze the data. So what that turns out is now you can analyze hundreds of thousands of protein sequences. With that amount of data, you really need some kind of machine learning. And because, like I mentioned, it's alphabets. So you need some kind of like a language model to analyze those. And that's how I got into the field. And I've been thinking about that for a long time. And in 2019, I think my idea is pretty mature. I applied for a grant within Genentech. There's a grant called Innovation Fund that you can propose an idea, they give you money, and then you can use 1 year to do your research. So I proposed my idea, and then I got an email after a few hours. They say, I'm sorry, you are not in the research team. So at the time, I was hired into another department, not part of the GRED. GRED is a research, Genentech's research. So I was in different department, but I still had this idea. So I applied, but they say, you are not in GRED, so you cannot apply for the fund. And I was a little angry, so I emailed the CEO, Bill Anderson at the time, say, hey, we're all within the same organization. We have great idea, and you should support that. And you shouldn't limit the idea just in the research department. So a few minutes later, I got an email, not from Bill, but from, I think, his secretary. He says, we will consider revising the policy next year, meaning they're not going to take me into the proposal. So that's part of the reason that I decided maybe I should build my own thing. And if I can build my own thing, this technology can benefit a lot of companies trying to do, develop a protein more efficiently. So I think that's the story.
Okay. So let's go back to your professional. For Leon, you've been working on immuno-oncology. Can you explain a little bit about what's immuno-oncology and what's the major factor you consider it is a good therapy?
Yeah, I think there's a lot to answer for that question. I will try to brief and also try to explain at a high level to allow the audience to understand. So immuno-oncology actually is a recent breakthrough in oncology treatments. So the traditional oncology treatment is target cancer cells, bring the cytotoxic warhead, including chemo or other target therapy, including kinase inhibitor, to kill the tumor cells specifically and directly. Immunotherapy is a different concept. Immunotherapy doesn't target tumor cells directly, but instead it targets immune cells. It uses immune cells as a pre-drug. Activate immune cell, allow the immune cell at a better capacity to control the tumors and achieve a durable response. So that's the basic concept. And the real revolution just happened 10 years ago, actually starting from Jim Allison's work, actually at Berkeley. At that time, he was at Berkeley, developed a CTLA-4 antibody. CTLA-4 is a break in T cells. So when T cell activated, there's a break to suppress activity, to prevent overactivation and autoimmune phenotype. And then Jim Allison identified that if you use an antibody to block the break, you can induce a much durable immune response, and T cell can be reinvigorated in the tumor environment and then kill tumors. And then with this effort, actually the first IO therapy got approved, which is anti-CTLA-4 antibody. And later on, there's another treatment being identified, which is PD-1, and identified by Japanese professor Huang Johnson. And then both Jim Allison and Huang Johnson actually win the Nobel Prize, I think due to their great contribution in identifying the treatment and develop IO therapies. And these are very exciting concept because we are not only tumors which there's a high heterogeneity in the tumor environment based on different indication, different individuals, based on different genetic backgrounds. So it's very complicated to treat, and also every patient has different response. But for immunotherapy, because you're not target tumor cells, you target immune cells. So what we found is that as long as you have sufficient neoantigen can be recognized by T cells in the tumor environments, there's a wide variety of tumor indication can be responded to immunotherapy. So the current PD-1 therapy has been approved in more than 10 solid tumor indication. Probably is the most indication approved for any cancer therapy in human history. And then CTLA-4 is the first one, but however, CTLA-4 has toxicity issue. Both CTLA-4 and PD-1 has toxicity issue, but CTLA-4 has more toxicity issue. So its application has been limited and has been replaced by anti-PD-1. So with those excitement now in industry, particularly in oncology discovery, actually a lot of focus has been on immuno-oncology and try to identify the next generation of immune checkpoint. Some targets similar like CTLA-4, PD-1. A lot of efforts still ongoing, but since after PD-1, it seems like there's lack of new identification of new checkpoint that can deliver such a promise as PD-1. And then now we rely on more comprehensive approach, including combinational strategy to enhance the immune response. But I will talk about the challenge later, but I would like to just highlight at a high level to summarize the current advance in concept of immunotherapy for my part.
So continue, I would like to know more about the challenge in IO field. What would you say? Like not every patient react the same, or there's something else?
Yeah, so the lesson we learned about the IO therapy, particularly for PD-1 or CTLA-4, is that there's a subset of patients. They have pre-existing CD8 T cells in the tumor environments, and those CD8 T cells already being activated to recognize certain immunogenic tumor antigen in the tumor environments. And the PD-1 antibody is particularly effective against those type of tumors. But however, those only account for a minor population of subset of patients in the clinic. For example, in lung cancer, only account for 20%. In other tumor types, could be even lower. So the real challenge is for the other 80% of the tumor, why the immunotherapy doesn't work, why PD-1 doesn't work. And then the major hurdle we identify so far is because this other tumor actually has much lower mutation burden. Because the more mutation you generate from the tumor, the higher chance your T cells are able to differentiate the tumor from your healthy normal tissue, so therefore can generate a more specific immune response to control the tumors. But if there is low mutation burden, your T cell has the last chance to recognize the mutated tumor cells. So for those tumor cells that unfortunately has low mutation burden or less immunogenic, the focus has been how to induce a more immunogenic peptide from the tumor cells, so therefore can train and educate our immune system, including T cell, to recognize and control the tumors. And that has been a challenge so far. And then there's a lot of efforts. So for example, there's some targets actually inside the tumor cells regulating the class 1 antigen presentation pathway. When inhibit, actually can allow a different diversity of peptide presented by the tumor cells, and therefore can be recognized by the T cells pool, and therefore initial immune response. But those are in the earlier discovery space. So hopefully there will be some advance and can further, and therefore allow a more durable immune response, cover more broader patients. And other challenge, I think Sony just touched base on the cell therapy. I think that's a very hot area in addition to the checkpoint blockade I just mentioned. Cell therapy actually provides a great efficacy and significant promise, particularly for the hematological tumors. And for CAR-T, for example, some tumor indication has been achieved almost more than 90% overall response rate, which is unheard of, unprecedented before. But however, people try to identify and then apply this amazing therapeutic platform into solid tumor, but so far it's not successful. And there are many reasons, because a lot of the tumor antigen we select also expressed in normal tissue, particularly for solid tumor. And because of the reason the CAR-T applied for solid tumor, the real challenge is the toxicity. It's difficult to overcome. You will achieve toxicity that the CAR-T2 eliminates or attack your normal tissue before you see the therapeutic efficacy to attack your tumor cells. And for the liquid tumor, this is achievable because your blood cells, including B cells or other myeloid cells, is dispensable. So even though you use those CAR-T to deplete your hematopoietic cells, the patient can still survive. But the patient cannot afford to survive if you attack normal tissue due to the overlapping of a TA expression between tumor and normal tissue. So I think that's the number one challenge that need to be overcome before we can develop more effective therapeutics for CAR-T in solid tumors. I will stop here, and then there's a lot to say, but we can follow up.
Okay. And for I-Chueh, you mentioned about gene delivery. So can you give us a high-level introduction of what gene therapy is doing and what major focus are you working on?
Okay, so currently we usually use the viral vector, so adeno-associated virus, which usually don't cause disease. And the lentiviral vector is for CAR-T therapy, so it's another kind of gene delivery into the cell. So currently, most of the gene therapy companies trying to do the systemic gene delivery. So AAV is commonly used, and so AAV is a small particle, and you can deliver about 4,700 base pair length of gene. So there is still size limitation, and so if patient has some genetic mutation, especially it's autosomal recessive, you can do the augmentation therapy. You just deliver the wild type gene, you probably can correct their gene deficient condition, and but still limitation if it's autosomal dominant disorder, so it will be very difficult to treat. So of course, this is for rare disease. There are a couple different companies, usually large pharma. They're already trying to stretch out to see whether they can use the gene delivery as a way to deliver drug, especially antibody drug. So just like diabetes retinopathy, usually use the anti-EGF antibody. So, but you need to do the frequent injection. So gene therapy can be a solution. So you just do one-time injection, and the drug can constantly express in your cell. So this is a condition, and I think this kind of application will be applied to multiple disease. And you don't need to go to the hospital so frequently. You just do one dose and potentially can last for your lifetime. So the thing I'm doing now is we're trying to develop different kind of platforms. So we modify the AAV. So as we know, AAV don't have a good specificity. They just go to different kind of cell randomly. So usually clinically, you need to use very high titer to achieve your therapeutic goal. So my job is trying to modify these AAV capsid and combine with other approach to increase the cell specificity. So we wanted the gene delivered to the target cell, not the bystander cell. So we probably can decrease this kind of dosing issue because high titer dose also correlate to the adverse reaction. So this is one thing we are working on. And another thing I'm working on is for mitigate immune response. As I mentioned, we use virus as a vector. So virus can trigger immune response just like SARS-CoV-2, that COVID-19 can trigger immune reaction. AAV can also trigger immune reaction. So it becomes a problem because some patients don't respond well. So for example, the Zolgensma, so the Novartis drug, they have 30% of the patients have poor responses. So you might want to consider the redosing. But because you already exposed to it, you already have the antibody. So the redosing efficacy will drop significantly. So that is the big issue in the gene therapy field. So a lot of companies trying to mitigate this kind of issue. So using this kind of approach, so such as immune suppressant, but the result doesn't seem promising. So all companies do have some in-house product. We are trying to see whether we can use in-house product to try to minimize this kind of condition.
So you already mentioned several challenges like dosage, efficacy, immune response. So how about, does it really go to the target site, those kinds of other challenges? Can you talk about that and how you improve that?
So as I mentioned, so most of the gene therapy are used to treat real disease. So for the company perspective, it doesn't bring too much money, and the therapy is very expensive. So most of the gene therapy product, you need to spend 1 to 2 million. So that will become a limitation. The patient population is too small. So that's why, just like our company, we're trying to try different way to just integrate like antibody therapy into our gene therapy delivery platform, and then we try to broaden the application in therapy. And another limitation, I think, is the gene size. So as I mentioned, for AAV, you can only do 4,700 base pair. So if your gene bigger, you cannot use AAV. You need to consider other approach. So there are a lot of different companies trying to use different things. Lipid nanoparticle is a pretty hard field now, but it's different. So it's not like Moderna or BNT vaccine. They deliver RNA. RNA don't trigger substantial immune reaction, especially innate immune. But for gene therapy, we want to deliver DNA because we want a long-term transgene expression. So this DNA material can trigger very strong innate immune reaction. And so there is still a predicament we need to overcome. And multi-gene disorder is another question. So just like diabetes, for example, if multiple gene is affected. So if you want to correct all these genes, later will be very difficult. You probably need a better vector to deliver multiple genes into the cell at once. So of course, a couple company trying to use the double AAV transduction or a different approach, but I don't think they are promising. So you probably need to consider to use other viral vectors. So adenoviral vector or herpes virus vector can be considered. You have way better capacity. But each viral vector has its own issue. So it's not an easy way you can fix all these problems. So yeah, and another thing I want to mention is just like our company, we currently focus on the CNS and brain gene therapy and eye gene therapy. So delivery is also a big problem. So for this gene therapy, you need to do a very invasive procedure. Yeah, to open the brain or to just inoculate the vector into the eye. So the way you can overcome this issue, you probably need to use a next generation delivery system. So this vector or this method material can cross the blood-brain barrier or blood-retina barrier. So you probably can do the systemic injection, and you don't need to take the invasive procedure. So yeah.
Thank you. And for Andrew, my question for you is that AI is such a new field. So how do you seek for resources, guidance, your challenges, and how do you know your model is doing in the right direction?
For the last answer, I think it's easier for me. Like my client's happy, I know it's going to the right direction. But the challenge, before I answer the challenge, let me describe what kind of, what are we learning here? So like I mentioned, protein sequence is a string of characters. It's actually a language of nature. There are certain keywords. For example, we talk a lot about antibody. We all do the COVID test at home, right? The kit has a lot of antibody that can recognize the COVID virus. That antibody is a sequence, depending on how you define antibody. It's around maybe 120 to 200 amino acid sequence. There's a certain keywords in that sequence that can recognize the virus. If you mutate the keyword out, you swap it, it won't recognize the virus. It will become a nonsense sentence, or it can maybe recognize something else. So definitely there's a language there. How do we learn that? So think about human learning language. If now I wanted to learn Korean, I probably would just go to Korea, or I just keep speaking Korean and then gauge people's feedback and figure out, and my brain will learn what are the words I say may be right, and I'll try another one again. Same thing in learning the protein sequence is that we will try a bunch of protein sequences. The way we speak protein is that we generate a lot of protein sequences through a lot of DNA synthesis technology like Linya's company is doing. We put into the cell to produce a protein. That's how we speak. And then to gauge the feedback is to measure the functions. That's the response from some assays. And then we use NGS to collect the data, to quantify them, and then use the language model, that's the brain, to process that. So back to the human language, in order to like OpenAI train ChatGPT, it's relatively easy compared to in the protein space because there are so many human language data on the internet. And also you can hire a lot of people to label the data for you because human language is created by human. So you can hire people to tell you, hey, this data means something. But the problem for the protein space is we don't understand the protein language. It's created by nature. We don't know the rule. We cannot hire people to label the data for us. So that's the challenge. And the other challenge, there's no internet for all this protein functional data. There's a lot of protein structure data, but what's important is the functions. There's no such data available on internet that you can do. So you need to generate that yourself, and that's what we're doing. Now, speaking of generating data to learn, the challenge there is people say garbage in, garbage out for machine learning. That's maybe easy for, not in like language, human language space, because a garbage sentence, we know the language, so we know this is not a good sentence to put into the model. But for protein sequence, we don't know what that means. We don't know what's garbage. That's a problem. In order to identify garbage in the protein universe, you need to set up some kind of QC matrix. And then in order to know what QC matrix you need to set up is that you need to understand the experiment. Because generating those protein require a lot of steps in the lab, and every process will introduce noise into the step. You really need to know how to do those experiments, what kind of noise can be introduced so that you can set up the QC matrix so that your training data will be high quality and your model will be robust. So those are the challenges and some kind of solution I'm suggesting.
Thank you. So that's the individual question, and I have a joint question for all of our speakers because our forum is focusing on AI. So the first question for Leon and I-Chueh is that have you been working internally or externally with AI or machine learning group? What do you expect that they can solve your current situation? And what's the gap between expectation and reality? Just like what Andrew said, the antibody after AI, it can have better binding, but not necessary function. So let's start from Leon.
Yeah, so I certainly can share AI perspective from big biopharma because, for example, Pfizer currently, we are very aggressively trying to adapt AI platform into our drug discovery and development pipeline. So currently, Pfizer even try to develop our own AI platform internally, which is different from GPT. Similar, but however, we cannot use GPT due to confidentiality information. Right? Pfizer have a lot of proprietary information that we cannot release in the public. So therefore, we cannot use public AI system to train the machine learning and acquire the information we would like to advance our drug development project. So we need to have our own proprietary AI platform internally, and only Pfizer project or scientists can allow to use that system. And then we just recently built that system internally. And the ambition is that, Pfizer generates so much data from pre-development, start from early discovery, target identification, validation, screening, all the way to phase 1, 2, 3 trial. It's a humongous database that if you can use AI properly, AI can help you to identify a relevant drug target pathway that gives you the highest possibility chance to be successful in the clinical development. Think about this, any single drug, it takes like at least 6 to 10 years to develop, to get FDA approval. And then companies need to invest billions of dollars, right? That's a huge investment. If we can use AI to help us to better to predict what drug give us most likely successful chance in the clinical development, that can save tremendous resource and also the timing to develop a drug, therefore can benefit more cancer patients. So I think there's a great potential layer that we can leverage. But however, the flip side is that we need to be careful about the security. And then the data cannot be shared between companies because they are competing each other. So we cannot share those information. So that's one thing that we are trying to do right now. Another thing we try to leverage AI is to identify novel PD marker. PD marker means pharmacological dynamic marker, which is a marker to predict the responsiveness of a single drug treatment. And then oftentimes we have a lot of new targets, new treatments, and then we don't know what will be the best PD marker to predict response. So therefore, we evaluate all the possible markers from immune cell. For example, there's 300 markers at different time points. So therefore, there's a humongous database will be generated to identify a more sensitive, robust PD we can use in the clinical development. So we plan to also use AI system to tell us that which biomarker but based on AI training can let us know we should follow up in the clinical development. So that's in progress right now. So hopefully AI can show more and more encouraging potential and help to shorten the time and the resource we need for our current drug development model.
Okay, so I can say 2 examples for gene therapy. So currently, one field is pretty hard is to do the capsid engineering. So there are a couple different companies just do the capsid engineering for the adeno-associated virus. So they just initial stage they just randomize the different sequence and trying to do some modification of AAV capsid. The goal is trying to find the capsid which can deliver to the specific target cell more efficient or more specific. So this kind of randomization, they probably can create millions of the sequence and the millions of different AAV. And, they do the in vivo screening in animals, so multiple rounds of in vivo screening. And each time they got the candidate, they can refine their model to create a new set of library to do this kind of screening. And this is pretty much like common practice. So Spark did it a lot. It's another gene therapy company. Our company just trying to do it, just stop because our gene therapy team just have 2 years. We just started gene therapy team 2 years ago. So this is one thing AI can help to shape what kind of capsid you try to use in the future. And I do think if we can combine with like structure-based design, might be even better. So a lot of like AI made the sequence, they just destroy the whole protein structure and the AAV become just useless. So if we can add another layer of consideration, which might just save a lot of time and efforts. Another thing I want to say is our J&J team, we do have an in silico team. They're trying to establish this immune modeling system. So they are currently focusing on the NHC class 2. So they want to predict certain immune epitope interacting with the T cell receptor. And so, of course, it's not only limited to gene therapy. They're also trying to do something about oncology. So our team idea is to, because the AAV, they do have specific immune epitope. So we're trying to do this kind of matching modeling system. So there will be specific HLA gene, just like specific, and then NHC class 1 or NHC class 2 gene, which might relate to stronger immune reaction. So in the future, possible ways, when you have a patient group, you can screen their immune gene, HLA gene, and then you potentially already can predict what kind of immune reaction may be happening in the future when they receive the AAV gene therapy. I do think there is a potential, but still a long way to go because it's just pretty early stage. Yeah.
And for Andrew, when you work with your clients, what's the major issue you always face? Is there a translational gap, knowledge gap, and how you usually solve this problem?
Right. I think there are a few, but I think the biggest one, especially early on, when AI wasn't that popular in the biotech space, 2019. So people think about AI as a black box. They don't trust the prediction. So they tend to not work with anything AI predicted. And that's also the same issue for me. I was the user of my product. So I actually spent a lot of time trying to see why the model makes such predictions. So I make it more explainable. And in order to make that more explainable is you have to open up the model, look at all the weights, and use some statistical method to interrogate all the weights. And then so I spent a lot of time making sure I can explain the model, the prediction to the client. I think that ended up helping they trust the model. And when they see a few prediction and the reason I explain to them, and if it's reasonable, they can go back and check the literature. Does my explanation consistent with the literature? Then they start trusting the system. So I think that's, the biggest issue early on.
Okay, thank you. We have 5 minutes left, so I have one last question. This is called Taiwan Tech Summit. So I wanted to link with Taiwan. So from your experience working in your field, do you have any pros and cons or experience to share when you collaborate with Taiwan or Asia? So we can start from I-Chueh. You're not the first one every time.
Not so much working with Taiwanese company. So I think it's difficult because the gene therapy currently is still focused on rare disease. And the market consideration and probably is the main reason prohibit a lot of people to invest in Taiwan for gene therapy. But I do think there should still be some opportunity. For example, if we can just use gene therapy to treat common disease, that can be a consideration. And just like I mentioned, you can do the antibody delivery, and then potentially can be a good start. And you don't need to worry too much about the patient population. And you can also make your system work well. And ultimately, probably can try to explore to treat a certain specific rare disease or multi-gene disorder. So that's my opinion.
Or Andrew?
Yeah, I actually thought about these questions and went to Taiwan the past 2 years and trying to talk to people, see if they're interested in applying AI in making protein products. But in terms of like applying AI in protein product, those are too new to them. And I think the reason is there's not enough application, and the reason there's not enough application is there's not enough data for people to try AI in the protein space other than the protein structure. So I think that's the challenge, but the solution I think is, if you don't know, Taiwan actually manufactures a lot of protein that go into clinical trial. So like people here, we develop protein candidate, outsource to Taiwan to manufacture that and then ship those to the hospital to do the trial. So that means they have the facility to culture cell to produce a lot of protein. That's a lot of data, but those data that they're currently generating are limited. Those are only for specific work that they're making, so it's not useful for machine learning. For machine learning, you need a diverse data set. So if we can teach them how to turn those engines to generate diverse protein, and then use next-gen sequencing. I think NGS is still not very popular in the protein space. It's very popular in other space, but not in the protein space. So if we can combine like how to generate diverse data, combine NGS, and then Taiwan has a lot of AI talent, we generate data set for them to make model, then we can leverage Taiwan's resources to become a data generation powerhouse and then build products. So, yeah.
Leon?
Yeah, so from Big Pharma's perspective, I do see now is a really good timing for Taiwan to develop biotech and pharma industry. I think one major reason is because Big Pharma nowadays, they invest much less than in their discovery because they realize, they spend too much money and it's just not worth it. I just using the Pfizer as an example. So their business model now will be that rely on external research discovery effort engine, and then they will invest and license those products and then move to Pfizer for phase 1 to 3 clinical development and become the products. And due to this reason, I do see a good opportunity for Taiwan because Taiwan wants to invest in drug industry, but however, Taiwan doesn't have the infrastructure, for example. It takes time to build infrastructure. And also it takes a lot of resource and money to build a big pharmaceutical company, which will take longer time. Pfizer exists for almost 200 years. If Taiwan want to do something, I don't think Taiwan has 200 years time to do that. So because of the discovery now, is less a focus in big pharma now. I think Taiwan is opportunity to change in that process and to become an engine for novel discovery science and target identification, and then provide the information that target validation for big pharma. So therefore, pharma can continue the pipeline to develop a drug. So it's like a whole process. We work together at the early upstream, and then big pharma work at the later downstream. I want to give an example. People like, especially people from Taiwan, like TSMC as an example, right? TSMC produce chip, but they don't produce iPhone. They don't produce Mac. But however, chip is so important. Every single Mac and iPhone need in the chip. So in Taiwan, it's the same. You could be the engine to identify novel targets. That's so important that all the big pharma want the targets, want to collaborate with you, want to know how you do that, how to identify that, and build that relationship. So, I think everyone here will agree with me that Taiwanese people are smart. I think no one will have any rejection here. So, we should focus and prioritize our resource into those novel target identification, which provide opportunity for sustainable long-term biotech or pharma discovery or development industry in Taiwan. I see that's opportunity right now.
Thank you for all the answers, and we finish this session. At 4:30, we have a networking time. If you have questions to ask the speakers, welcome to join us. And especially I-Chueh, he's from J&J. You can know what's up with the vaccine at that session. Thank you.